Okay, so it's 2 p.m. If you can hear me, as always, raise the hand or let me know it's working. Okay, so it's working, so it's good. If you notice about the schedule, as you see from the Slack channel, we get the replacement video on YouTube last Monday, and that's covering the two sessions. One is the session 5 and 1, genetic architecture, and then the first part of the session 5 and 2, and that was the constitution of the last Monday class. I hope everyone gets that video for your lecture, and we're going to finish off the session 5 and 2, which is the remaining part of the experimental design today. And then this will be the last lecture on this semester, up to the session 5. And we are scheduled to have the review and the Q&A section this Monday, but I noticed that the next Monday will be the exam period, so probably students will be on the preparation. So I will replace that time with the Slack channel. This is our the typical activities. So come to the Slack and leave your questions, then we can discuss on that. And some students already started, preparing their exams, as I see in the channel. And then we'll have the exam on June 21, which is the Monday after the week, right? So that will be the I think that will be the most suitable time for your exam preparation. Otherwise, it would be too early for your preparation. So I set up the week after the next week. Okay, so we're back to the session, which is the method for the association testing. So before we going into this topic, the first thing you have to understand is we have wide range of genetic architecture for the traits and disease. So if you look at certain trait or disease, there's no way you can understand in a unified perspective. So there are various ways you can understand for the genetic vectors. So for instance, if you look at the patients with developmental disorders, they are mostly affected by de novo germline mutation, right? And if you look at patients with cancers, and those people are mostly affected by somatic mutations, right? And if you look at certain conditions like Marfan syndrome, these are affected by rare inherited variants. Or if you look at Alzheimer patients, or if you look at the patients with Parkinson's disease, those people are common inherited variants. So the point I'm talking this here is not just the memorizing which disorders affected by what. This is the situation you if you are interested in certain disorder, then that disorder should have affected by certain genetic factors, and that factors can be various by each disorder. So let's say if you are interested in type 2 diabetes, and type 2 diabetes probably affected by mostly by common variants, and some affected by rare variants, and few people probably affected by developmental disorders. So likewise, if you are interested in certain disorder, then you have to look up their genetic background and genetic architecture. That is the first thing in the modern research. And if you are interested in certain factors, and that genetic factor is also part of different methodologies. So if you are interested in de novo mutation, then you have to use certain technology. And if you are interested in common variants, then you have to use the different technologies. So I can categorize into the few testing. So the first one is the the testing based on locus. So we call the per locus test because you are testing association based on locus, right? So in that case, you can compare between the the case and control. So especially when you analyze the common variant or the rare variants inherited from your parents, and those kinds of the genetic factors, you are using the per locus test, right? But then if you are comparing the de novo mutation, then de novo mutation, you can think of their perspective. You can think of their properties, right? De novo mutation are not likely occurred in a same locus, right? De novo mutation are random mutation. They are randomly occurring in our genome. So there is a almost zero chance to have the same mutation at the same locus, right? So in this case, you have to use the different testing unit. So this case, we use the per gene test, right? So we're not gonna, there's no way we can compare the case and control about the de novo mutation on the same locus. So you have to compare the number of de novo mutation per gene, right? So that's why we shortly call it per gene test, right? Another way we can compare the per category test is you comparing the number of mutations between case and control by category. So gene could be one category, but you can set up the different definition for the category. So if you, let's say, if you want to look at certain transcription factor binding sites, then in that case you are refining the testing unit into the genomic region of transcription factor binding sites. Then you can compare the number of mutation between case and control, right? So per gene test or per category test is basically use the characteristics of variants that are not occurring at the same sites because of their rarity, right? So de novo mutation is one case, and rare inherited variant is another case because the rare inherited variant is very low chance of having the mutation occurrence at the same sites. So that case also use the per gene or per category test, right? So likewise, if you look at each individual genetic factors and their association into the research, then you have to pick up the proper methodology for your research, and that research also evolved over the time in a different context, right? So why I'm telling this situation to students? The reason is that everyone talks about the big data. Everyone talks about whatever the data science is critical in a future biological research, but surprisingly, not many people are talking about the methodology because the methodology is the key things in a the future application. Whatever you want to do research in future, or whatever you want to do, become a clinician in a future, or if you want to work in an industry, in any other situation, understanding methodology is the key because genetic factors has been studied in a different context for different traits and disorders, right? So this is the sort of the starting point. You can think about, which kinds of methodology or the experiments you used in a different research situation. Okay, so first application, which is first methodology, which is the per locus test. So we are commonly call this method as the genome-wide association study, or shortly GWAS, right? So GWAS is typically looking at the association of common variants, right? So common variants is by definition, this is the very commonly seen in a population, right? And common variants, as we learned in a session 2, these are working as a additive factors, right? They are contributing very small percentage of the contribution to total traits, right? So if you want to look at the association of common variants, then you're basically looking at the number of variants happening at the same locus in a case and control, right? So if you look at the SNP1, you measure how many times the cases have this genotype, right? So you collected 2,000 people as a cases, and out of these 2,000, you have 1,200 people, right? And compared to cases, you have 1,000 people of controls having this genotype, right? So you can basically use the binomial testing, then you can calculate whether this number differences is significant or not, right? Then you can calculate the p-value, whether that is the strong association or not, or you can calculate the odds ratio as well. So, and if you look at the second SNP, and second SNP has the cases versus control, it's like 1,020 versus 1,000. So it's almost same, right? So we are not seeing very significant association with this SNP, right? And third SNP is the same situation because the cases versus control is almost the same, ratio. It's almost close to 1-to-1. So it's unlikely we can say that this SNP is associated with the disease, right? So per locus test is basically comparing the lots of numbers of SNPs in our genome. And as we learned from the session 3, the common variance is representing the certain haplotype. So we don't have to look at all the common variants in a haplotype, right? So what you have to use the tag SNPs because tag SNPs representing the haplotypes, right? So you can pick up the tag SNPs, then you basically comparing the number of certain genotype between case and control. Then you can test that whether that difference is significant or not. Then in this case, you're not comparing just one SNP. You're basically comparing the lots of SNPs, like hundred thousands or millions SNPs, right? So basically, you are holding many hypotheses. So that's why we call it many hypotheses in this approach. In other words, we call it multiple hypotheses. So what you have to do is you have to set up the different threshold to choose the real genome association. So in that case, we use the multiple correction, and I will deal this issue in a later section. Okay, so common variant, we can use the SNP microarray or whole genome sequencing because basically, if you can read any the text names across the haplotype, then either microarray or whole genome sequencing can work, right? This is one of the revolutionized study in 2014, and this is the GWAS of schizophrenia, right? So GWAS is actually first introduced in 2006, which is about 15 years ago, and that time GWAS wasn't that successful. So the first GWAS at the time in 2006 was the age-related muscular degeneration, which is the sort of the eye disorder, and that was successful. So that time, people used the 80 patients or something, and they did the GWAS and they find some mutation. And people was quite excited about that because people thought that if you use the GWAS, then we can find the mutation, we can find the gene associated with the disorder, and we can conquer the disorder, we can develop the drugs, we can develop the treatment. But other than this GWAS, the eye disorder GWAS, most of GWAS was failed, right? So people was, doubt on the GWAS research, and people, claim that, GWAS not gonna work in a biological research, right? And surprisingly, to date, some people still think in this way. So, some biological researchers, they think, GWAS is totally failed research and it's not going to be valid anymore. But it's not true. It's totally not true because lots of large, the big pharmaceutical companies like, Pfizer's or, Genentech or Amgen's, like, the leading companies in the world, they are focusing on GWAS. The reason is that GWAS, they are actively sharing the data set, they're actively developing the methodology, and their research quality was totally changed after the 2015 14 or 15 these days. And this paper from the Nature in 2014 was one of the starting of the revolution. So schizophrenia GWAS, they collected 36,000 cases diagnosed for schizophrenia, and they collected 100,000 control samples, right? And after this GWAS, they basically comparing the 100,000 SNPs in the genome, and they comparing the number of genotypes that have very huge differences between cases and control. And what they found 108 loci significantly largely observed in schizophrenia cases, right? And one of the genomic loci at the time was the very, the most strongest association at the time was the HLA region on chromosome 6, and this region containing the gene called complement 4, which is the C4 gene. And this complement 4, what these genes is doing, they are working in an immune system, right? And people thought about that, immune system in schizophrenia doesn't really make sense, because schizophrenia is the mental disorder, and this is something wrong with the the brain issue, right? The synapse or whatever, the cognition issue. So what are these immune system genes are doing in this schizophrenia? But it's actually true, because the brain are not just a part of the synapse. The brain has the immune cells, especially microglia or astrocytes, and those immune-related cells in our brain is critical for the brain development. So if you're losing the controls of these immune cells in our brain, then your brain is overgrowth and overly complicated, and that is actually changing your mental states as well. So schizophrenia related to this synaptic growth synaptic pruning by the immune system gene was proven as a correct hypothesis after this research, and lots of people continue to research the immune system affecting on schizophrenia and other neurological disorders, right? And this is one of the hot topics. Lots of people study in these days, and also especially like microglia or astrocytes are likely be sex biased in terms of why schizophrenia is more commonly observed in males compared to females, and they are thinking about why female brains are differently developed compared to male brain. And one of the strong hypothesis is the microglia hypothesis, and this is also originates from this GWAS research. Okay, so back to the data set. So if you look at the lines here, represented by red color, and this is the level of significance. So if you are remembering the statistical testing, we are normally use the p-value, the threshold 0.05, right? So 0.05 is the threshold whether you call significant or not. We have much more philosophical way why significance, threshold working in this way, but let's focus on the convention way. So we are focusing on 0.05, right? But as I mentioned that we are using the multiple hypotheses for your testing, right? Because we are using large number of text SNPs from our haplotypes, right? So we are using 100,000 SNPs. So we basically divided by the threshold by the number of the hypotheses you used in your testing, right? So that's why your threshold for the significance is getting lower than just 0.05. So you basically divide by 100,000 numbers. So it's going to be 5 times 10 to minus 8, right? And you are, if you see the locus above this threshold line, then you call it significant, right? And then if you look at the plots, there are lots of diamonds here, and these are the index SNPs, or you can call it tag SNPs. They are basically same. And then those are, and above this line and the diamond, the SNPs on here, they are the significant SNPs used find from this study, right? So this is the way you basically representing the result of the GWAS plot. And typically we call these types of plot called Manhattan plot. I don't know why people name in this way, but I mean, people remind of the buildings in Manhattan in New York, and people call sort of these types of plots is a Manhattan plot. And on x-axis is the order of the chromosomal position. So from the chromosome 1 to X and Y, but normally Y is omitted because it doesn't exist in female. So we normally comparing the exist one in both sexes. And y-axis is the significance of association, which is the minus log 10 p-value, right? So we can basically showing the how much each SNPs are associated with the disorder. Okay, so GWAS study, we can also apply this concept for the rare variant, right? And rare variants, we can also use the per locus test. So basically following the same notion. So if you look at the first SNP, then you can compare the numbers of the genotype between the case and control. So here you see the case is 18 and control is 1. Then you put it in a binomial testing, calculate the odds ratio, then you can think about whether this mutation is associated with cases or not. But then problem is that the rare variant is rarely exists in a population compared to common variants. So you have less chance of having the same locus in a cases and controls. So you're basically dealing with the lower numbers compared to common variants. And also another point is that you have to compare much larger number of loci compared to common variants. So think about the research from 1000 Genome Project or HEMMET project. You see the number of common variants from the population or the number of rare variants from the population. We have seen much larger number of rare variants in our population because the rare variants is mostly uniquely exist in a individual. So if you collect all of the loci from the each individual, then you will essentially see larger number of the loci from the rare variant part, right? So in other words, you basically have much larger number of hypotheses. And also if you are seeing the proper test, then you have to use the larger number of samples, right? And if you want to do the rare variants, you can basically use the whole genome sequencing or whole exome sequencing, right? You cannot do it by microarray because microarray is designed based on the common variant. Okay, and this is the examples of the Manhattan plot of the type diabetes. So the plots on the above is the common variants and the below one is the rare variants. So you're basically comparing the differences in terms of the threshold and from the multiple comparison, and also the number of loci associated with the disorder as well. Okay. And also another point is that, the examples of type diabetes in the same disorders can be affected by either common variants or rare variants, or the disorder can be affected by both rare variants and common variants, right? So even if it's the same disorder, the constitution is actually contained different parts from the different genetic factors. And this is one of the key points in these days of the genetics, because, in old days, if you look at certain disease, you think about only one types of genetic factors, right? Like, this disorder probably affected by only common variants or by rare variants, but it's actually wrong. If we study more and more, if we look at the data and more data set, then we see, lots of combinations between the rare variants, common variants, somatic mutation, and de novo mutation. So you have to see the points that, even if we see the same disorder, the disorder can be, affected by many different genetic factors. Okay, and another types of the testing methodology is the purging test. So as I mentioned that this is the testing the number of variants from the cases and controls, and you collapse the numbers within the same gene, right? So in this case, your testing unit is not SNP, it's not locus. This is the by gene, right? So if you set up the gene, then you count the number of variants from the cases and control. So if you look at the gene A, that has the 2 mutation from cases and 2 mutation from control, 0 mutation from controls. And if you look at the gene B, the same number of mutation from cases and controls, right? So why we have to do this way? Because there is no way we can set up the same locus to compare the cases and control, because the mutation we are using in this concept are so rare, right? So you have to think about, different ways of the testing. And another consideration in this approach, you have to think about the length of gene and also sequencing quality, right? So if you look at the gene B and gene C, gene B is longer than gene C. So there are, your expectation is gene B should have more mutation than gene C. So you have to adjust the length of the gene in this experiment. And also another aspect is that you have to think about the sequencing quality. So all of the data set in a gene B in this area has to be sequenced in all samples, either in cases or controls, right? So if you look at the gene A, you see 2 mutation from cases and 0 mutation from controls, right? Then you can probably conclude that, cases has, gene A probably the cause of this gene, cause of this disorder, because we see 2 mutation from cases and no mutation from controls. But in certain situations, you might have the data set like, there is no sequencing data on a controls because, the sequencing was poorly performed. So you didn't get any, data from the controls, and you get the data from cases. So in this case, there is no way you can check whether the controls have the mutation in this region or not, right? So counting the number of variants in each gene, you have to consider sequencing depth as well. And in this experiment, if you do the multiple comparison, then you basically use the number of genes in human genome. So one example is this is the number of de novo mutation from autism cases compared to unaffected controls, right? And you basically count the number of de novo mutation happening cases versus controls, right? Then you calculate the statistics out of that. And if you probably remind your first assignment, the Kaplani's paper, and that is also use the same the purging test for their association testing, right? And you can recheck whether the Kaplani's paper has the the appropriate for the locus test or the purging test. Then you probably feel that the purging test was the correct way to get their significance of association. Okay, so last methodology is the per category test, and this is mostly used for the ultra rare variants. And in this case, we're not counting the number of variants per gene. This is the number of variants per category, right? So, category can be defined in a arbitrary way. So you can make a category for the variant consequence, right? Like non-synonymous mutation or synonymous mutation or protein truncate mutation. Or if you're looking at the mutation from the non-coding genome, then you can basically set up the category for promoters, enhancers, or introns or intergenic regions. So you can basically set up the different category by your definition, right? But then critical things is why we need to set up the category for our testing. So the first reason is that if you have the too small number of samples, but you want to do some, experimental analysis for the association, then in that case, you can use the category-based testing. So let's say if you have 100 samples for your de novo mutation research, then you want to compare the numbers between the cases and controls. So the first thing you can ask is whether de novo mutation is mostly appeared as a missense mutation or appeared as a protein truncate mutation. So this is the first way you can refine which genetic factors are the things you have to look at first, right? So this case, you can use the category test. Or the variants of your interest, like a de novo mutation, are various parts in our genome. In this case, category test is way efficient way than locus test. Or if you don't have the specific testing unit like gene, right? So if you think about the non-coding genome doesn't have the concept of the gene. So there is no way you can refine the testing unit. So in this case, you have to set up your own testing unit for the testing, right? So this is one example of the, again, autism exome. So, so this study from the 2012, right? So compared to this study, this is from 2019, right? So it's 7 years ago. This 2012 paper is one of the revolutionized paper in the genomics field, because, I mean, this is the first large-scale genomic research, not just autism, in other human disorder as well. And what they found is they found a strong association of the de novo mutation, especially in a nonsense mutation. And this paper is the, okay, let's look at the paper. This paper published in Nature in 2012 was great paper. What they found is that they found the de novo nonsense mutation strongly associated with autism cases. And other than this paper, oops, not good. Yeah, this is the Sanders 2012 paper. It's cited more than, I mean, it's about 2,000 times. And this is the one paper. I'm just introducing the one examples, but not the paper coming in a same day in a same Nature paper. So this is from the Nature on a April 4th, right? And at the same time, this is another paper in the same day, April 4th. And another paper, rock. Another paper from the Nature 2, and it's published in November, but it actually first published in April, right? So April 4, looking at it here. So why I call this paper is a revolutionized. The reason is that there are almost zero chance to have replicate the results between studies, right? And that is the first thing. And another thing is, at the time in 2012, we didn't know anything about autism at the time. And people was curious about, autism can be, I mean, some people argue that autism, caused by the cold mother, refrigerator mother. And the reason is that your mother doesn't give that much affection to child, because of the child get autism in their childhood. There are many spurious hypotheses at the time, or other people argue that vaccines probably, caused the autism. Those, hypotheses was totally wrong, right? So what people found in these days, as I mentioned, the de novo mutation and nonsense mutation is highly associated with autism patients. And surprisingly, not just one study found that result in a Nature in April 2012. The three papers are indicating the same result, and they're coming in a same day and same journal. And given the the history of biological research, these things is almost extremely rarely happening in our field. And because of this, the starting point, people, start think about, the de novo mutation is doing something in autism or other developmental disorder, and people start extensively studying the cause of the de novo mutation in a childhood disorders and also related neurological disorders as well. So setting up the very robust and very good experimental design and finding the good things at the early times, it can change totally the notion in our field. So that's why they introduced the research on here. And at the time, this study doesn't have that many number of samples, right? So they are basically comparing the three categories. So nonsense, missense, and silent mutation, right? And then mutation is the, those types of the research is based on the category test. And that time, the association of the nonsense mutation was totally effective. What effective means that was totally correct. And that research is replicated by other studies. And also, more recently in 2019, they improved their experimental design. Then now they are looking at the gene level association, and now people can say 100 genes are significantly associated with autism spectrum disorders, right? And these things are totally revolutionized the notion of the disease research. And this is another category based testing. This is for the de novo mutation, but in a non-coding genome. And this is published in Science in 2018. It's actually by me. And this is your second assignment. And what you have to understand is why we are looking at the category in these types of research. The reason is that non-coding genome, as I explained, there are no way to make a definition of the gene in the non-coding genome. So you have to set up the different ways of the testing unit, and non-coding mutation, you have to set up the certain way of the category for the association testing. So what the study does at the time was creating the arbitrary category out of the pre-existing information for the non-coding genome, and using this number of category and doing the association testing as a as you put it in the coding genome equivalent testing, right? And this is another, the very early days of the sort of the non-coding research. And, as we see in a Sanders paper in 2012, there are lots of room to improve the research of the time. But the thing is that the notion of these types of the association methodology has to, evolve from the category test to the per gene test, or in different sections, we are using the locus test and do sort of the large scale genetic associations testing in the real world field. Okay, so if you do the genetic association testing, the things you have to consider in your experiment is the confounders, right? So what is the confounders? So what you have to do in a genetic association testing, you're basically looking at the patient's diagnosed for a certain disorder, then you find the genetic cause. But the problem is that between these links, sometimes the hidden variables can change, can distort your association, right? So we call these variables are confounders, right? So confounders is a variable influence both dependent variables or independent variables, right? So dependent variables is your genetic cause, and independent variables is your disorders, right? So you are comparing the the certain disorders, but sometimes it can be distorted by sort of these variables, right? So one famous example is the population stratification, right? So if you want to, let's imagine that if you want to compare the Alzheimer disease, right? So you collect the cases, 1,000 cases and 1,000 controls. then you compare the common variance between cases and controls, right? And you find some gene, and you call that this gene is associated with Alzheimer. Then boom, it's great because you found the gene. Then you develop the drugs. Then you develop the treatment. But that can be wrong. So one wrong situation, one wrong experimental design like this. So if you collect the samples, 1,000 samples from the Asian, and you collect the 1,000 samples from the Europeans, then you compare the cases versus control for the Alzheimer genotype, then it's wrong. The reason is that common variance is heavily influenced by population stratification. So that's why we are dealing that stratification population structure in a session 3, right? So if you want to do really good experimental design, then you have to collect the samples from the Asian or from the Asian for the cases and controls, right? If you mixed up, different ancestry for cases and control, then your association testing will be distorted, right? And another, the advice for your career. So, some people probably argue that, why we are learning this, population stratification or population structure for our genetic class, because, some students might argue that, these things are too hard for the, the second year, genetics, but it's actually wrong. The reason is that, these days, lots of companies or even clinicians, they want to use the the genetic research for their treatment or drug development, right? But the point is that they are in their university days, they are mostly learning the Mendelian genetics. So there's no way they have no chance to learn about the population structure or population stratification. So what they cause is that there are no idea how to design the experimental design for the common variant or the rare range, right? So this is totally awkward for the the population stratification can be confound your association testing. That's why I keep emphasizing that the session 3 is the critical for the clinical research as well, because lots of students probably wants to be in a biomedical science. So the things like population stratification is the key things we are really care about the confounders in a genetic association, especially for the common variants and rare variants. So you have to always do the quality check for your population structure in your data set. Then you apply those information into your experimental design, right? So this is the one confounders. And if you study de novo mutation, then you have to look up paternal age, because de novo mutation are naturally occurring by advanced paternal age. So old fathers are likely to have more de novo mutation because there are more chance of having replication errors in a germline generation, right? So this case, you also have to adjust those numbers in your experiment, right? So this is one key example, or you can remove those confounding factors, right? So what people do is people looking at how much these variables, these confounders are interact with your number of genotypes, right? So you basically do some exploratory analysis to looking at sort of this information. And we are covering more on the hands-on or practical, experiments in a biostatistics course in a second semester. And also, as I mentioned, that if you find those confounders, you can basically remove or adjust the numbers by cases and controls. So if you didn't, adjust the numbers, then you see the differences, naturally exist in a cases and controls in your data set. So you have to adjust the paternal age between the case and control. Then you see there is no effect from the paternal age. Then you can find the true signal from the biological factors, right? So this is the thing you have to keep in the experimental design. Okay, another important consideration in an experimental design is the understanding the differences between the meta and mega study, right? So what is the meta-analysis? Meta-analysis is the combining the summary results from the multiple studies for the sample phenotype of interest, right? And meta-analysis is basically provide a very efficient and practical strategy for overcoming the limitation of the power. And you can basically, you see more results from these the meta-analysis. So what I want to tell you is, so let's say you do the autism exome sequencing result and your friends did the same thing and your father did the same thing. So basically the three data set exist in near you, right? So you find the number of mutations from cases and control, and your friends did the same, your father did the same thing, right? But different samples. So if you want to increase the statistical power, you basically collect all of the samples from the three people and into the same data set, right? Then you recalculate the significant and odds ratio, then see whether this gene is associated with the disorder or not. The reason is that more samples gives you more confidence, right? So that's why the meta-analysis is one of the simple things you can try in your data set, right? But then the mega study is a little bit different. It's actually quite different, right? So what mega study did? Mega study is they collecting all the data set and collecting all of the raw data set. So the thing about the meta-analysis is only collecting the number of de novo variants from each study, right? But mega study is you basically collect all of the raw data, which is the FASTA-Q files, right? Then you collect all the FASTA-Q files from 3 studies, right? Other studies, and then you process this data set using the unified pipeline. This pipeline means that, from the FASTA-Q, alignment, BAM, and VCF, right? So you basically use the same pipeline, then you can ensure that the confidence and reproducibility of your research, right? So this is the basically very strong point of the mega study. And also you can basically control the confounders inherent in a different data set, right? So each data set probably has the different the quality, and this situation you can also controlling that information too. But then problem is the problem of the mega analysis is that this is very expensive because you have to pay additional cost, right? So you basically pay, collecting raw data from others, right? Because you have to, pay for the data transfer, like, you have to buy the, disk space or the computational server, then you have to spend your money on the data processing, right? Or sometimes the raw data set are missing, right? Or not accessible, right? So these things are the disadvantage of the mega analysis. So what you have to do is for the meta and mega analysis is think about some studies you learned over the time, right? So think about the study of the Kaplanis in your first assignment. Then ask yourself, Kaplani's paper is the meta-analysis or mega-analysis, right? If you read the paper, then you will find that Kaplani's paper is either just collecting the numbers from different data set, or they basically get the raw data and they process in the same pipeline, then do the, the unified processing for the entire data set. So you can basically see the differences between meta and mega. And also assignment 2, you're looking at the science paper annotated, right? And you can also see that whether this paper using the meta analysis or mega analysis as well, right? And also you probably remember the NOMAD database we launched in a session 5 and 1. and you can see that whether that database is created by meta-analysis or mega-analysis, right? And one spoiler for the NOMA dataset, this is the mega-analysis, and this is the strong point of why NOMA dataset is so valuable in our field, because they are doing sort of the mega-analysis in the field, right? And this is the one point you have to understand the meaning of the meta and mega analysis in the real world research. And also just advice for your career. I mean, I highly recommend you to be engaged in the team, whatever research or industry are doing mega analysis, right? I mean, as I mentioned, the mega analysis is expensive and it costs extra, you know, the labors on the data analysis, but mega analysis is much more valuable than meta analysis, right? And this is one trend. Lots of institute, lots of the industry, they want to do mega analysis, right? So one example is we are mentioning many times. This is the UK Biobank data set, right? So this is one of the biggest data set created from the United Kingdom, the British people, right? And this is like a more than 10 years of data set, right? And I mean, basically the participant from the, you know, the UK area, right? And UK government invest the money for the data set, right? But surprisingly, this data set, half of this data set is basically you know, funded by the big pharmaceutical company, right? So companies are helping their project. The reason is that companies, you know, I mean, think about the reason. So UK Biobank is the public data set. So once the data set is created, it's open for everybody. So I can see that, you can see that, your friends see that, anyone in the world can see the data set. But why big pharmaceutical companies, they want to fund the data set, and why they want to pay their money to this project? The reason is that they pay money and they get the data set way earlier than others, way earlier than publics, right? So they want to get the raw data and they want to process in a mega way, and they get the valuable genes or valuable association, valuable data set finding from the UK Biobank cohort, right? So what I can give you an advice is that you know, working in a, you know, the organization doing the mega analysis is way better than just doing the meta analysis, right? Meta analysis is easy, and it's you know, it can be operated by very soft money, right? But mega analysis is really serious business and much more, you know, the hard standard required for the, you know, researchers or the industry workers, but it's very, I mean, it's very worth, right? So it's, I mean, if you are considering a career in a data science or the, you know, the genomic research in your future, then be in a group doing mega analysis, and also you know, grab a chance to do some mega analysis in your career time. So that's why I, you know, introduced the two different concepts between the meta and mega analysis in more recent trend. Okay, so another consideration for the experimental design is the reproducibility, right? So what is the reproducibility? This is the independent researchers arriving at the same research using their own data and methods. So basically, if you do something and if you try to reproduce something that has been done by others, then you basically see the same thing. So this case, we call that there is a reproducibility, right? And replicability refers to a different teams arriving at the same research using the original authors and authors artifacts, right? So replicates is also quite similar way the describing you see the same research from the experiments done by others, right? So this reproducibility or replicability is the critical things people wants to wants to improve in these days experimental design. This is another sort of philosophical debates between the researchers, but think about 80s or 90s biological research. The beauty of the research in early days is to try to find something at the earliest, right? So if you find something new, which hasn't discovered by others, then people thought about high value because it has the novelty, right? But the value of novelty is still something in these days, but the value of reproducibility is much more considerable in these days. So people start caring about the reproduce of the something research or the result is the good things or not, right? So in 2008, Nature, they write lots of the editorials or the papers of the reproducibility, because lots of people see irreproducible results from the individual studies, right? So yeah, if you have the time, yeah, you can read it. Why I introduced this? It's a little bit tricky to say, but some of you might want to do research in future, right? And you are being in a team doing some research, right? Research team, basically. And sometimes professors or PI, your mentor, ask you to do some research. And sometimes your research from the research is not something you are expecting or your professors is expecting, right? And sometimes very bad situation is your mentor asks you to do again or try to manipulate that result, right? These things is really toxic. These things are really bad. But lots of the situation, lots of the the environment, lots of the cultures in a the research field in industry as well, they are not really transparent in terms of the reproducibility and those types of the philosophy, right? So one of the things we experienced in our society was the one of the drugs not reproduced in other experiments, and this is done by the large company in Korea, and that was about the muscular the disease related drug development. And why these things happen in our field? Because people are not really critically evaluate whether your experiment is reproduced or not, and people didn't see really the value of the reproducibility in the research, right? So if you are really think about the Korea in a research field, and not just the academia and also industry as well, you have to get a sense that choosing the team really caring about the reproducibility and transparent discussion and also the communication as well. So again, this is why I ask you to do some cultivating your communication skills on Slack, because if you after your graduation, if you became a workers in industry or a research team, then you are the one part of the members in the organization, and you have to raise your voice and contribute to the discussion and think about, critically evaluate what you guys teams, done in the project, right? But especially in our culture, the discussions or debates is rarely happening because of this stupid hierarchical cultures, right? So even in a university, we are not cultivating how to give a question. We are memorizing things. You are just following what the professor is doing, right? So we are living in a different society. We're living in a different times. We have to know about how to discuss and how to bring the issues, how to bring the agendas to the points that critically evaluate your research and interpret the experiments you've done in your time, right? So one of the aspects we really care about in these days is the reproducibility, right? So think about this, and also I highly recommend you read this article. It's not going to appear in the exam, so not going to be rushed in a 2 weeks, but over the vacation, then this is worth reading in your time, because knowing about the reproducibility or not will change your career totally, right? So that's why I introduced that things in here. And another critical part I introduced here is the gene, right? So one gene we know about the depression is the serotonin. The serotonin is not the gene. This is the neurotransmitter, and this is in brain. And first reported in 1950, about half centuries ago. And so serotonin became a hypothesis for the psychiatric condition, especially depression, right? And people try to find the genotypes and mutations in a serotonin transporter gene, which is the intaking the serotonin and delivering in a different part of the brain, right? And that function is the operated by this gene SLC6A4, right? And this is the one of the common genes. People are heavily studied for the depression. So what people do is they creating the mice model because they want to validate this mutation in a depression, right? So they choose this hypothesis for the mice model, and people heavily study this disorder, right? And if you read some, the media or the blog post, people are quite talking about depression and serotonin gene, right? So serotonin gene is the cause of the depression, and these types of, the character is always in the blog, right? But surprisingly, we do the large-scale genomic research, and we never see replication of the serotonin receptors in a large meta-analysis. Serotonin receptor is not causal genes for the depression, right? This is surprising things, right? So why these things happening, right? So the first thing is, at the time, people didn't use multiple correction in their experiments, and people didn't care about the population stratification, right? Another reason is that they choose the hypothesis at the first time, serotonin gene, and they keep focusing on the gene. They didn't take up other genes for their hypothesis testing. So there's a publication bias, right? And also early studies, like in these days, like the 90s or 2012, at the time, we didn't have enough sample size, right? So these things are not replicated. So why I'm telling this issue, the reason is that if you want to become a biomedical researcher in some days, like a graduate schools or in your career, choosing a gene is like a choosing partner for your life, right? This is like a marriage. This is like a wedding, right? You choose one gene, then you study that gene for your entire life. But what if your choosing gene is wrong, right? This is the tricky thing, right? Because there are a couple of examples. This one gene is found in one family in 2001. This is very famous schizophrenia gene. Over the 20 years, people study this one in schizophrenia patients. But after 10 years, this one gene is not replicated at all in a large scale samples. You see one mutation in one family, But if you study 10,000 samples, but not see association. But some people doing this quant research over the time, they finish PhD, they get a job, but it's not replicated. So what can we do? So knowing about the reproducibility and replication in a large scale research, this is why this really matters in your career time. And another region is the Wiener curve. This is the originally from economic field, but this is also commonly used in the genetic field, right? And what Wiener curve, a newly identified genetic association in an initial study is always overestimated when the statistical power of the original study is not sufficient, right? So if you look at this mutation in type 2 diabetes, this is very famous mutation and people want to develop the treatment drugs for the type 2 diabetes patients. And if you measure the odds ratio in a 91 samples in 1980, in 1998, there was the odds ratio was 4, right? And after 2 years, people looking at 3,000 samples, which is way larger sample size than 91, and the odds ratio was 1.3, right? It's decreased. And after the decade, we're looking at 13,000 samples, and this mutation odds ratio is only 1.1, right? So the first estimation of the odds ratio in early days was overestimated, right? And these things also happen in autism research as well. So if you look at the nonsense mutation, odds ratio was around 5. And these days, if you measure the odds ratio of nonsense mutation, this is about 1.5, right? So much more realistic values, right? Okay, so that is the one thing about the reproducibility. And one thing why we care about the experimental design is the multiple correction. So as I explained that, if you use the large number of hypotheses in your experiments, then you're basically controlling your threshold for the significance, right? Because your association testing can be wrong, right? So this is the one reason. So that's why we basically divide the number of the test you used in the experiments, then you set up the new threshold for your testing. So this is basically the way we are applying the multiple hypotheses, and I explained in a previous slide. And challenges in association testing. This is sort of some remaining issues in our experimental design. So the first issue is the heterogeneity, right? So if you look at the same, the disorder, we basically collect large number of samples, but each patient probably have different trait values. So in other words, we can say that phenotypic heterogeneity might exist in samples, right? So sometimes, doctors can be misclassified, or sometimes, the measurement is wrong, or sometimes, the disorders is natively, the heterogeneous in a population. Or sometimes the patient can have different, the comorbid condition, which is, bipolar disorder patients can be also diagnosed for the depression. So all of these phenotypic heterogeneity can be a problem in a association testing. So what people want to do is to do more rigorous clinical ascertainment in a phenotyping. So if you do the genetic research for the major depression disorder, you can do clinical evaluation by doctors, or you can do by questionnaires, right? So of course, by doctors is much more objective and much more rigorous way to ascertainment. Then if you do in this way, then you will see more, less heterogeneous samples and get more homogeneous population for your association testing, right? So doing the very detailed phenotyping is really highly, perceived in these days, because, phenotyping is much more expensive and also much more labor intensive. So if you are keen on the biomedical research, the improving the phenotyping way is another very promising the field you might want to pursue. And also the genetic heterogeneity also exists in a disorder as well. Okay, so what I want to tell you is the phenotyping can be advanced by the technology. So I imagine that some students might have the wearables like an Apple Watch, and these things collecting your health information like the heartbeats or heart rates or the oxidative rates from your vessels or your sleep patterns. Why Apple's or why those companies are doing this? Because they want to collect the information and they want to relate that information to the genetic factors as well. And one example is like a 23andMe. 23andMe is one of the large genomic companies in US, and they basically collect the genetic information and also the health-related questionnaires, and they are doing some, the genetic research based on their information. So this is again can be done by the online participant because everyone in the world can do these types of questionnaires when they are connected to internet, right? So this is way efficient way than visiting doctors at the clinic, right? So this is the one way people are doing in these days. And another problem is the genetic heterogeneity. So genetic heterogeneity is that if you look at certain disorders, that disorder can be affected by many numbers of genes, right? So one example is again autism. So if you look at the autism patients, as we see in a previous slide, we have 102 genes associated with autism, right? So surprisingly, all the patients diagnosed for autism, but their causal genes, so their genetic problems are different, right? So some patients have the mutation in a gene 1, some patients have the mutation in gene 2, but they are somehow converged as a autism patient, right? So what people want to care is that, not the same, the disorder is not always developed by the same gene. It can be developed by different types of the genes, right? So this case, we describe the situation genetic heterogeneity. And another way of the genetic challenges is the pleiotropy. So pleiotropy is meaning that the single gene can do multiple functions. So why single gene can do multiple functions? The reason is that the single gene can do multiple functions, then it's good for the evolutionary perspective, because in your developmental situation, you have to cope with the various the situations, right? Like you have to differentiate into the different cell types or different environmental factors affecting on your development, right? So in each different situations, you have to use the different cards. You have to use the different, the items to overcome the situation. So the easiest way the biological organisms can do is they're using the same gene in a different situation, right? This is much more efficient way you can cope with that different situation. Also, in a highly evolutionary organisms, they are using these pleiotropic genes. So why do we care about this situation? So pleiotropic gene is, you have the mutation in a gene 1, then mutation can cause autism, but it can cause microcephaly, which is the your head circumference, head size is way larger than general population. So that is the phenotype macrocephaly. Or the gene mutation, this gene 1 mutation can cause seizures, right? So think about the situation. If you are working in a clinic, right? You are doing the genetic testing, right? And you find the mutation in a patient, and the mutation occur in a gene. And that gene's mutation can be associated with autism, can be associated with seizures, can be associated with microcephaly. So which diagnosis term you will give to the patient, right? So this is another challenge in a clinical situation, and people wants to develop much more accurate algorithm to prioritize the very good estimation from the pleiotropic gene background, right? So these things are like a pleiotropic or the heterogeneity or the phenotyping issue is the current remaining challenges, and in your the ongoing career, you might want to participate and pursue over the time. Okay, so there was the very big rush and the long presentation about the session 5 and 2. So hope you enjoy the sort of the latest sort of the backgrounds of the experimental design. And I mean, as again, the session 4, session 5, the the items and topics in this here is quite new to you, but again, this is very basic for your starting point of your learning, because I mean, somehow the latest biomedical research is quite advanced and complicated over the time, and it's very fastly evolved over every year. So you have to follow up the latest technology and latest topics, then you can survive in the the cutting edge field. So hope you enjoy the session 5 and 1 stuff and 2 stuffs as well, and review your stuff, and always highly encourage leave your comments or questions on Slack. And this is really the end of the semester, and hope you really enjoy the genetic class of the semester. And we are moving on to the real world data set in a biostatistics course in the second semester, because I mean, knowing the hands-on experience on the real world field will be really great benefit for your career. So that's why I'm preparing the latest topics for you in the second semester. So hopefully you enjoy the very end of the the semester time, and hope you do the good preparation, and hope you continue to discuss the latest items in second semester. That is my very last wish for you. Okay, so all things good, and see you on 21 June for your final exam, and also keep discussing on Slack.